All right, welcome everybody. Today we're going to be looking at the structure of DNA. And DNA is pretty sweet because everything that we know about it has been discovered very recently. So it's a lot of new information, new discoveries in science. So what does DNA stand for? It's deoxyribonucleic acid. So it's nucleic acid found within the nucleus of eukaryotic cells. If you remember prokaryotes, it's actually a picture of a prokaryotic cell. Prokaryotes, the DNA is circular and it's just floating around in the cytoplasm. And the shape of DNA is a double helix that looks like a spiral staircase. So it's got the sugars on the outside of it and base pairs in the middle that look like the steps of a ladder or of that spiral staircase. This picture over here is an electron microscope picture of DNA. You can see it kind of all swirling around in the cell. So DNA has three important roles. The first is to store genetic information. So it's basically the blueprint for all cells. And it's able to keep that information over a long period of time so that the cell can keep producing the proteins, producing the things that it needs for the cell to live and for that organism to keep going on living. It also allows for the copying of genetic information. So DNA can be copied and sent out to other parts of the cell, to the ribosomes, for making protein. And it's also for transmitting genetic information. So if we didn't have DNA, we wouldn't have something that could be passed from parent to offspring and continue on a species. If you look here on the right, this is an example of DNA showing its components. We have the sugar phosphate backbone. This part winds around on the outside. Then, and the sugar is it's deoxyribosugar, so that gets the D in the DNA name. In the sugar, this picture doesn't show it, but connecting each one of the sugars to each other in the backbone is a phosphate group. And so that's why over here it's a sugar phosphate backbone. They alternate between a sugar, sugar, phosphate, wrapping around here. And then on the other side here is the sugar phosphate. And then in between, we have four different bases that are used in DNA. Adenine, thymine, guanine, and cytosine. And the base pairs line up in the middle of DNA. Those are the letter code that code for proteins. Okay, so now I'm going to have you take a second right now. I want to pause the slide and I want you to draw out a nucleotide. So a nucleotide is the building block of DNA. It has a phosphate group here, sugar, and the base pair. So one way to think of it in a really simplified form, just think of it like a house. So your deoxyribosugar is your house. Coming off your house, you've got a chimney. So the phosphate group is your chimney. And the base pair is kind of like the garage of the house coming off the side. And each nucleotide is linked together, forming a chain, and that's what would be the outside, that sugar phosphate backbone on the outside of your helix, and on the center is your base pair. Okay, so DNA bases. Adenine and guanine are purine bases. Right here, they're two rings. And cytosine and thymine are pyrimidine bases. These are single-ringed molecules. Uracil here, we're going to talk about this later when we talk about RNA, so don't worry about that now. Just focus on cytosine and thymine. Now, to make it a little bit confusing, purines match up to pyrimidines, and A and T bond together with two hydrogen bonds. So here's our T and our A coming together. Two hydrogen bonds linking them together. G and C bond together with 3 hydrogen bonds. These weak hydrogen bonds hold the DNA molecule together at the center. This is also, the hydrogen bonds can be opened up. So zipped and unzipped allows DNA to open up to allow for copying. And so they're just the weak hydrogen bonds. If if a DNA molecule has more guanine and cytosine, so let's say it's 80% guanine and cytosine, it's going to be held together more tightly at the center. It's going to be more difficult to break that DNA molecule apart than if it's mostly made up of adenine and thymine, where it would only have 2 hydrogen bonds. Okay, so if you know adenine, then you know the amount of thymine. Because these two pair up together, if you have DNA strand that is found to have 5% adenine, would have 30% thymine. Now, that would give you 70% of the base pairs of your DNA molecule. So if you think about it, if it has 15% guanine, then it's going to have 15% cytosine. And that would be 100% of your base pairs. If it was 45% adenine, some questions might ask you. So if you could think about if you have 45% adenine, how much guanine and cytosine would you have? And then also try if you had 20% adenine, how much guanine would you have? So you can try a couple of these practice problems, write them down into your notes, and then bring them into class. So DNA is a coded molecule, and it can be read and rewritten into different forms. A, T, C, and G are the genetic alphabet. They're written as a code, and they can code for proteins. When read, they translate into instructions on how to make proteins or enzymes. So DNA is the instruction manual to make proteins. DNA is like the underlying blueprint of an organism. And that's it for today. Really want to focus in on understanding the structure of a nucleotide, the overall shape of DNA, and then also be able to tell which base pairs are going to pair with each other, because we'll do word problems on those in class.